My name is Isabella Hoffbauer, and the date today is September 17th, 2020. Today I will be doing a presentation on the determination of gender, genes versus hormones. First, I thought it would be beneficial to give some background of the sex hormones in males and females. They're very similar, but also there are some differences. So there are three main structures of the female and male sex hormones. There is the gonads, the internal genitalia, and the external genitalia. The gonads produce gametes, which are the cells produced that create new human beings when combined together. In females, the gonads are the ovaries, which produce ova, which are the eggs, the gametes. In males, the gonads are the testes, which produce sperm. Those would be the gametes. The internal genitalia are accessory glands and ducts that connect the gonads to the outside environment. The external genitalia are all the structures for reproduction that are external on the body. It is important to note that sexual development is programmed by the human genome. Almost all nucleated cells in the body consist of 46 chromosomes, which is a diploid number. Diploid just means that they come in 22 matched or homologous pairs of autosomes plus one pair of sex chromosomes. Autosome is a word to describe chromosomes that are relevant for development but not sex differentiation. So it can do with development but just not the sex differentiation part. Diploid cells are written in 2N, which just indicates pairs. On the other hand, the only cells in the body that don't consist of 46 chromosomes are the egg and sperm cells, which only consist of 23 chromosomes. So these chromosomes are haploid, which means they are not matched, and they are written in 1N, meaning that they are not in pairs, so they are not homologous. The point of these cells being 23 chromosomes is very important, and it's because the egg and the sperm cells are supposed to combine to create 46. When this happens, this is called a zygote. Now we're going to talk about sex chromosomes and how they determine genetic sex. So, the sex chromosomes consist of X and Y. A female parent has two X chromosomes, and a male parent has an X and a Y chromosome. So each egg cell in the female has an X, and the sperm cells either consist of an X or a Y. When two X's join together, the sex of the embryo will become female. If an X and a Y join together, the sex of the embryo will become male. Depending on whether an X and an X join together or an X and a Y join together, this determines what the external and internal genitalia will develop into in the embryo. There is also a possibility that sex chromosomes can be abnormally distributed during the time of fertilization. So, for example, if there is multiple X's, but there's also a Y, this embryo will become male, like I have indicated on the screen that XXY can be male. This is because if that Y is inherited, it doesn't matter if there is two X's, it will become male. If only a Y is inherited, so no X, that would be YO, and this embryo will die because the X chromosome is larger and has essential genes that the Y chromosome does not. So if only a Y chromosome is inherited, the embryo cannot survive. If there is only an X received, this would be XO, and this embryo would develop into a female, but this would be a genetic disorder because the Y is missing, and this is called Turner syndrome. So this picture is a diagram taken from the Silverthorne textbook, and it's basically just describing what I just said. So the female parent has two X's, and both eggs are X and X. The male parent has an X and a Y chromosome, and the sperm cells can either be X or Y. The two X's coming together create a female offspring, and the coming together of the X and the Y become a male offspring. So sexual differentiation, when a woman first gets pregnant, it is impossible to tell the gender right away. And this is because the development of the reproduction organs starts in the 7th week of gestation. So before differentiation happens, the tissues that the reproduction organs are made up of are called bipotential, because they cannot yet be identified. In the embryo, there is a bipotential gonad that will consist of either it consists of outer cortex and an inner medulla. If the embryo is female, the cortex will develop into ovarian tissue. If the embryo is male, the medulla will develop into the testes. The bipotential internal genitalia consist of the Wolffian ducts and the Mullerian ducts, and depending on the gender of the embryo, one duct will degenerate and the other will continue developing, which I will talk about in a second here. So, in the male embryonic development, which I personally think is a little bit more complicated than the female embryonic development, so as I already mentioned, if the embryo is male, the medulla of the embryonic, the medulla of the bipotential gonad will develop into the testes. This is caused by the SRY gene that produces a protein called the testes determining hormone, or TDF. And this binds to DNA to activate the genes SOX9, WT1, the Wilms tumor protein, and SF1, which is the steroidogenic factor. It is very important to know that testosterone, even though it is a male sex hormone, is not part of the testicular development. Like I said, even though that is a male sex hormone, but it is important later on after differentiation happens. So after differentiation happens, there are 3 hormones that will begin to be secreted. So the Sertoli cells secrete anti-Mullerian hormone, which causes the Mullerian duct to degenerate. Interstitial Leydig cells secrete androgens, which are testosterone. Now testosterone is being a part of the process, and dihydrotestosterone, or DHT. Testosterone causes the Wolffian ducts to continue to develop into the male accessory reproduction structures, and the remaining sex characteristics, such as external genitalia, are controlled by DHT mainly. So next we will talk about the female embryonic development, the last slide of my presentation. So in females, there is no SRY gene. So if there's no SRY gene, none of these genes, SOX9, WT1, or SF1, are going to be produced or acted upon. So since there's no SRY gene, the cortex of the bipotential gonads develop into the ovarian tissue, and since there is no anti-Müllerian hormone, the Müllerian ducts develop into the vagina, uterus, and fallopian tubes. And with the absence of testosterone, the Wolffian ducts degenerate, and then without the DHT, female external genitalia develop in place. And so that is basically an overview of how sex differentiation happens between genes and hormones. These are my sources. Thank you for listening.